Hello biology lovers, welcome back to SLD Biology. Today in this video lesson you'll be looking at digestive enzymes. So if you haven't subscribed already, please do so. Alright, in this video lesson you will mainly deal with the amylases and its associated gland, its site of action and the pH. Regarding proteases and lipases, we will discuss in the next video lesson, okay? I want you to pause it and go through the objectives. I want you to pause and imagine a life without digestive enzymes. Pause this video and give yourself some time and think about how your life would be without digestive enzymes. So you see, without digestive enzymes, the food you had remains undigested. It will just sit there in your stomach. And no matter how much you eat, you will starve. No matter how full your stomach is, you will keep on starving. Your body is unable to gain energy or nutrients from the food that you had just consumed. So that will make you starve, that will make you feel weak and tired. So you see, the digestive enzymes, they are the hidden heroes. They break down large, complex food into simplest, usable and absorbable forms. You won't be able to, your body cells won't be able to use this, but your body cells can use this for energy, for repair, for growth, for survival. And what do you need for that? You need digestive enzymes. So with this, let's get down with the three categories of digestive enzymes. All right, let's get down with the three different types of enzymes we need to associate with, which are amylases, proteases, and lipases. Amylases, they act on carbohydrates, such as starch. So the complex forms are broken down into simplest form. You see, this indicates chemical bonds, which bonds all these units and they make it complex. But when these chemical bonds are broken, they get converted into simplest form. Here you are dealing with the carbohydrate. So carbohydrates are broken down into simplest form sugar, which is glucose, by the enzyme amylases. Next we have proteases. Protease, they break down proteins. So the substrate here is proteins. Proteins, the chemical bonds in the complex proteins are broken down by the protease and they are converted into simplest form, such as amino acids. And lipases, what they do is they act on fats by breaking the chemical bonds here in the complex form. They convert it into simplest form, which is fatty acids and glycerol. So basically, these three enzymes, they act on these three substrates, which are carbohydrates, proteins, and fats, and they are converted into simplest forms, which are very small in size, which are soluble and can be absorbed easily by our cells for repair, for growth, for survival, for energy. You see? So this is the intro overview on the three types of enzymes. Now we will get down with the different types of enzymes found within each enzymes, okay? Let's get done with the details now. And the very first carbohydrate digesting enzyme you're going to deal with is salivary amylase, which can also be called as ptyalin. P is silent, all right? There you go. Salivary glands, which are exocrine glands because they have ducts, and whatever they produce in the saliva, they go through the duct and then they get into your mouth. and it helps in digestion. It helps in moistening the food and protect our oral cavity from the bacteria. Okay, there are three major pairs of salivary glands, which are parotid gland, and this is submandibular gland, and then you have sublingual gland. You see, parotid gland is located in front of the ears, and submandibular gland is located just below our jaw, isn't it? And sublingual gland is located under the tongue. Imagine there's a tongue here and it's just located under the tongue. And all these glands of salivary glands, they possess ducts. That's why they are exocrine glands. I'm sure you have learned this in your lower class. Exocrine meaning they possess duct, and whatever they secrete, they are poured into the mouth via their ducts. Okay, and one important thing about parotid gland is it is the largest salivary gland that produces the most salivary amylases, which means it produces the most saliva. So imagine if you remove parotid gland, how do you think it will affect digestion? Pause it and take some time to think. Yes, definitely, if you remove parotid gland, your saliva production will reduce, and if you align it with the salivary amylase, the salivary amylase will be produced in a very less amount, which means the carbohydrate digestion in your mouth will be affected. Not only that, your mouth will dry up, chewing and swallowing will become difficult. All right, you may lose a sense of taste, and it may increase the risk of dental problems. So you see, it's very important to take care of our salivary glands. Let us now discuss the action of salivary amylase. They act on starch, and then they break these chemical bonds, and these are converted into some into shorter polysaccharides and some into maltose. And maltose, as you can see here, there are 2 units, so it can be considered as disaccharides. Di means 2, and then saccharides means sugar. So some are converted into maltose, and some they remain as shorter polysaccharides. For example, when you chew bread very slowly and properly, you tend to taste it sweet, isn't it? The reason is because the salivary amylase in your mouth, what they do is they digest starch. They digest starch into maltose, which is made of 2 glucose units, and this maltose gives the taste, sweet taste. All right. If you analyze this graph, on the y-axis we have enzyme activity, and on the x-axis we have pH, and pH ranking from 1 to 6, it's considered as acidic, and pH ranking from 8 to 12, it is considered as basic. Now you can ignore pepsin and arginase for a while and associate only with salivary amylase, and let's see at what pH salivary amylase functions best. Okay, if you look at the peak of the salivary amylase, it falls at pH around 6.8 to 7, which means salivary amylase functions best at slightly acidic to neutral. So the optimum is around 6.8 to 7. And at this pH range, salivary amylase they can efficiently break down carbohydrates into simple sugars such as maltose. And when you swallow salivary amylase, it goes into your stomach. And you know that your stomach is highly acidic. And there's no activity of salivary amylase in the acidic medium. And why is that? Because your salivary amylase gets denatured, it loses its shape and functions, and therefore it is unable to catalyze the reaction, which means it's unable to break down the carbohydrates which are there in your stomach. Before we move on with the pancreatic amylase, I want you to grab a pen and a paper and answer this question. Why there is no carbohydrate digestion in the stomach? Pause the video and answer it. Alright, if you have answered already, there's no carbohydrate digestion in the stomach because, as discussed, the stomach is highly acidic. There you go, the stomach is highly acidic. And salivary amylase, they gets denatured, which means they loses their shape and functions, and they cannot act on starch. And that's why there's no carbohydrate digestion in the stomach. It's actually sad, isn't it? Next enzyme we are going to discuss is pancreatic amylase, which is secreted by the pancreas and pancreas. Now, one thing I want you to know here is, before you get down with the action of pancreatic amylase, you should be thorough with the anatomy of pancreas. So let's get down with the anatomy. Pancreas, it is an elongated organ and it's divided into 4 parts: head, neck, body and tail, and it is retroperitoneal, meaning it is behind the peritoneum. Now, if you have no idea on peritoneum, you can pause the video and browse it. And pancreas, they contain duct which runs along the length of the organ, and the duct, we call it pancreatic duct. Okay, and this pancreatic duct, it will join the common bile duct. You see, pancreatic duct joins the common bile duct. Common bile duct is formed by the fusion of hepatic duct, that is liver duct, and cystic duct, that is gallbladder duct. So the cystic duct, CYSTIC, cystic duct and hepatic duct, they join and form common bile duct, and the common bile duct and pancreatic duct, they join and form ampulla of Vater, which can also be written as hepatopancreatic duct. And now hepatopancreatic duct, finally it opens into the duodenum, and there is a round muscle around this opening which we call sphincter of Oddi. Okay, so the sphincter of Oddi, which is a muscular valve, it will control the flow of bile from the common bile duct and pancreatic juice from the pancreatic duct. So it will control the flow of bile and pancreatic juice into the duodenum. Not only that, it will also prevent the contents of the duodenum back into the liver or pancreas. So which means it's very important to have functional sphincter of Oddi to prevent the backflow of your duodenum contents. Otherwise, it will cause infection. So it can cause severe abdominal pain, nausea, vomiting, and it will also end up causing pancreatitis, which is inflammation of pancreas. So it's very important to have functional sphincter of Oddi. Next thing you should know is pancreas can have both the function of endocrine and exocrine. Endocrine, they are mainly associated with the production of hormones, Whereas exocrine, they are mainly associated with the production of digestive enzymes. So as an endocrine, our pancreas will have a group of cells, okay? Group of cells sitting right in the middle, which looks like an island. And these are actually made up of groups of cells known as delta cell, known as beta cell, alpha cell, okay? And these cells are responsible for production of hormones. So you see the islets of Langerhans are a small group of cells found in the pancreas that function as an endocrine gland, meaning they are ductlets. Whatever they secrete, they are directly poured into the blood, which means the hormones produced by these cells, they are poured directly into the blood, no duct. Alpha cell represented by this dark blue, they secrete a hormone called glucagon. And you know that glucagon will increase the sugar level in the blood, isn't it? And the beta cell represented by these light blue, they secrete a hormone called insulin. And insulin are very important for lowering the blood sugar level. And then there is delta cell, which secretes hormone somatostatin. And there is pancreatic polypeptide cell, also known as PP cell, that will secrete pancreatic polypeptide hormone. So these part islets of Langerhans, which are made up of these groups of different cells responsible for the production of hormones directly into the blood, they function as an endocrine gland. So which means pancreas are capable of functioning like an endocrine gland. Not only that, pancreas, they can also function as an exocrine gland, meaning as an exocrine part, they will have a cell known as acinar cells. They will have such many cells, acinar cells. And these acinar cells, remember, they will surround these group of cells, collectively known as islets of Langerhans. So these acinar cells, they will have a duct, as you can see here, right in the middle. Acinar cells, they have a duct, and they are responsible for secreting varieties of enzymes. Let us have a look at the enzymes. All these enzymes, pancreatic lipases, pancreatic amylase, chymotrypsin, trypsin, carboxypeptidase, nucleases, these are all secreted by the acinar cells, and then they get into the duct and they travel via the duct, and then they are drained into the duodenum. So whatever digestive enzymes secreted by pancreas, they are drained into the duodenum, which means they function in the duodenum. All right. Before we discuss the action of pancreatic amylase, I want you to take a screenshot of these questions. These are scenario-based questions, and answer it with honesty. Do not copy it from your friends. You put some effort, do some research, and answer the questions in your own language. All right? The pancreatic amylase, it will break down the starch into its simplest form, that is maltose, and some will remain as shorter polysaccharides. So what they do is they break down these chemical bonds and convert most of the starch complex form into maltose. All right, and the best pH for pancreatic amylase to function effectively is in an alkaline medium, that is around pH 7.1 to 8.2. And the reason why our duodenum is alkaline is because of the secretion of bicarbonate ions from the pancreas. Remember we discussed acinar cells? These acinar cells, not only they're responsible for secreting digestive enzyme, but they will also secrete bicarbonate ions, sodium bicarbonates, into the duodenum. So this sodium bicarbonate, they will neutralize the acidity which is coming from the stomach. So the food coming from the stomach, which we call it chyme, will be neutralized by the sodium bicarbonates. And the primary source of sodium bicarbonates is pancreas. Remember that, okay? Again, I want you to take a screenshot of this and go through. So what would happen without sodium bicarbonates? Without sodium bicarbonates, the acidic chyme, which is a partially digested food mixed with stomach acid, it's coming right from your stomach and it's entering the duodenum. So without sodium bicarbonates, you won't be able to neutralize the acidic chyme. Sodium bicarbonate, it is an alkaline substance that neutralizes stomach acid, which means it will raise the pH, which means it will make the pH alkaline in the duodenum, which is suitable for digestive enzyme activity, that is for protein digesting enzymes and as well as for pancreatic amylase. How will it affect carbohydrate digestion in the duodenum? Carbohydrate digestion in the duodenum, it relies on the enzyme pancreatic amylase. We have discussed that, isn't it? And this pancreatic amylase, it works best in slightly alkaline environment, that is around pH 7 to 8.5. So if there is no sodium bicarbonate, the acidic chyme won't be neutralized, which means our duodenum will remain acidic, and pancreatic amylase, they don't work in acidic medium, they become inactive and they will work inefficiently. And as a result, the breakdown of complex carbohydrates into simple sugar is affected, which means impaired. So this will lead to poor absorption of carbohydrates and also lead to some gastrointestinal symptoms like bloating, gas, and diarrhea. So what is the primary source of sodium bicarbonates? The primary source of sodium bicarbonate in the digestive system is, of course, pancreas. We have seen that in the pancreas there is a cell called acinar cells which will secrete bicarbonate-rich fluid rich with sodium bicarbonates into the duodenum via the pancreatic duct. So one more thing you should keep in mind is the release of sodium bicarbonate is stimulated by the hormone called secretin, and it is released when the acidic chyme enters the duodenum. So you see, when the acidic food enters your duodenum, what happens is the cells from your small intestine, they secrete a hormone called secretin, and this secretin will in turn stimulate your pancreas to increase the secretion of sodium bicarbonates, and that's how they reach your duodenum and neutralize the acidic chyme. So to summarize, without sodium bicarbonate, our duodenum stays too acidic. Pancreatic amylase activity is inhibited because it cannot function in the acidic medium. It has to be alkaline. Carbohydrate digestion is impaired. And the pancreas is the main source of sodium bicarbonate that helps maintain the proper pH of duodenum for not only for carbohydrate digestion but also for fat and protein digestion, which we will discuss in the later video lesson. Okay, let us now discuss the action of disaccharidases, which are produced by a cell called enterocyte. Now, where are these enterocytes present? If you see the lumen of our intestines, they are lined with finger-like projections called villi. And remember, villi are further lined with microvilli, and they appear like a brush, isn't it? You see? So these are the cells within the villi, and since they are lined with microvilli, they appear like a brush. And these cells are responsible for producing enzymes such as disaccharidases. The disaccharidase we have here is maltase. Maltase, they will digest the maltose and it will convert it into 2 units of glucose. Next we have is lactase. Okay, lactase will act on a milk sugar called lactose, and when the lactose is digested by the lactase, it will convert it into glucose and galactose. Next we have is sucrase. Sucrase, it will act on sucrose, which is a disaccharide, and it's converted into monosaccharide, that is glucose and fructose. Take these as a bloodstream and take these as an enterocyte, which are found within the villi. And villi, remember, it lines the lumen of our small intestines, right, and large intestines as well. Right now we are with the small intestine. We have moved on from the duodenum. We are now with the, let's say, jejunum, and now in the jejunum, these disaccharidases, they will act on their respective substrates. Maltase will digest maltose into glucose and glucose. Sucrase will digest sucrose into glucose and fructose. The bond is broken here. So sucrose is made up of 2 different units, that is glucose and fructose. And when it comes to lactose, which is a milk sugar commonly found in dairy products, it is again made up of 2 different units, that is galactose and glucose. So when the bond is broken by the lactase, it will be digested into galactose and glucose. Now these monomers, they are in simplest units, they can be easily absorbed into our bloodstream. That's where the absorption comes in, okay? You see, 90% of African and Asian populations, they have lactose intolerance, which means you don't have enough lactase enzyme in your intestine to digest milk sugar effectively. So lactose are basically found in dairy products, so don't take dairy products if you're lactose intolerant. So to summarize, lactose intolerance is a common digestive issue where our body cannot properly digest lactose, which is the main sugar found in the milk and dairy products. Normally, our intestine produces an enzyme called lactase, which can break down the lactose into glucose and galactose, so our body can absorb it. But as an adult, 75% they are lactose intolerant, which means we are not able to produce enough lactase. So therefore, the undigested lactose, which passes into the large intestine, which we call colon, these are fermented by the bacteria which are residing there in our large intestine. And when they do that, when they ferment it, they cause symptoms such as bloating, gas, diarrhea, and stomach cramps. And sometimes you even feel nauseated. So I want you to take a moment and take a screenshot of this and answer the questions through thorough research, okay? You know what? I really don't believe in taking dairy products. Because you see, I think humans are the only species that takes another animal's milk. Because dairy products and milk, including milk, they are heavily commercialized 10,000 years ago, supported by the government, heavily marketed through industrialization, through magazines, through TV, because it benefited them. But scientifically, if you go at a deeper level, these dairy products, meat-based products, animal-based products, they will only end up affecting your health. They will increase the risk of your cancer. They will increase the risk of cardiovascular diseases. They contain a lot of saturated fats in it. Your lifespan will become shorter. So I want you to consider all these negative effects, do thorough research, and answer the questions provided here. Okay? Getting down with the isomaltose, these disaccharidases, it will digest isomaltose into glucose and glucose. And the source remains same, that is brush border, which is the enterocytes. So we have reached to the end of this video lesson. I want you to take a screenshot of these questions and answer it in your notebook. And you can send the answers to me and I will do the correction. Okay? I hope you have done the screenshot already. Okay. So in the next lesson, we will discuss the proteases, which is protein digesting enzymes.